Hi class, welcome to chapter 8. This is on the nervous system. We're going to go through the whole chapter. All right, let's begin first by just discussing the necessary anatomy that we need to understand. So in the nervous system, there are two cell categories. There is the neuron, which is actually one type of cell, versus the glial cells. And the glial cells are a category of cells with different functions, right? Why would we have different cells if they didn't serve different functions? As you know, the central nervous system includes the brain and the spinal cord, but we also need to acknowledge the peripheral nervous system as well as the enteric nervous system. So the peripheral nervous system includes all of the sensory nerves that are present throughout our body, and they're primarily within the protected layers of the dermis. Oh yes, so when I cut my finger, here you go, it's almost healed. When I cut my finger, I slice through some of the sensory nerves, so it feels very weird at the tip of my finger. I can't quite feel that sensation because the sensory nerves are affected. So they send the signal into the nervous system to process a response. So every reflex that goes through the nervous system involves two different sets of nerves. We have the afferent nerves as well as the efferent, beginning with the E there, the efferent nerves. So the afferent nerves are the sensory nerves throughout our body that perceive different things, and the efferent neurons are going to respond by coordinating a muscular response, like to move the finger away, or but we also have the autonomic divisions. So a lot of different divisions in the nervous system. Even within our autonomic nervous system, that's divided into the sympathetic and the parasympathetic branches, and those are essentially opposite responses. We'll look at several examples. And then, as I mentioned a moment ago, we've got the enteric nervous system. And really, this was it's a recently discovered and appreciated area of the nervous system, also of the immune system. It's only really been in the past, I'd say 20 years or so, that researchers have begun to investigate the role of the nervous system and immune system in the gut. But there's a ton of neurons that are present in the gut, so they're there for a reason. These do fall under the control of the autonomic nervous system, but they're able to function on their own. So not every response goes into the central nervous system. That would be a good time to bring this up now. A reflex does. An example of a reflex is touching something hot and perceiving that pain with the senses, and then retracting to prevent further damage. But not every response goes through there. I mean, our heart is a great example of that. The heart does not involve the nervous system to generate the rhythm. It can be affected by the nervous system. Okay, so these are several of the different terms that are important to understand what we're talking about, to understand the vocabulary here. And we'll go through most of these as we go through, but now you have a list of different terms, as well as some of the other words that they may be called by. It's never simple with scientific words. All right, so this is a rather complicated figure because it summarizes all of the aspects of the nervous system. So seated within the brain here and the spinal cord is the central nervous system. It is composed of just those two organs, the brain and the spinal cord. But we also have the autonomic control, which is divided into the sympathetic and parasympathetic responses. So an example of a sympathetic response would be an increase in heart rate, where a parasympathetic parasympathetic response would be the decrease in heart rate. So examples of other tissues and responses that are under the control of the autonomic division are the smooth muscle. So that's lines our intestines, really lines all of those internal tubes in our body, but also different exocrine glands and endocrine glands, which are associated with the production of different hormones, and even some of the adipose tissue. We've got our somatic motor response, which is always going to involve the skeletal muscles. And then over here on the left side of the figure, let's run through from the very beginning. Let's apply this pathway now that we've seen several times in our lecture slides. So here we begin with a signal that is perceived by a sensory receptor. So in this case, we'll just say it's hearing because it's going right near where the ear is. So the sensory receptors pick up that sense and send it through the afferent pathway into what is called the integrating center. That is the central nervous system, and that's where it's processed. So the response includes the afferent path, which is complicated here. We've got the motor response, the skeletal muscle response, and then as we just went through, that autonomic response. All of this is happening also with communication with the nervous tissue and cells that are present in the enteric nervous system. And we've got pretty elaborate feedback responses. They're very simplified just in this pathway, but we'll come to learn and study some of those as we move through. So looking here with a question, we're looking at the autonomic division of the nervous system. What does it contain? We've got motor neurons, sensory neurons under voluntary control, or are both A and C correct here? So we're looking at the autonomic division. So let's go right here, division of the nervous system. It has nothing to do with motor neurons. So A, we can eliminate right away. So that also eliminates choice D since A is there. So now the somatic motor response is a separate effector response. Effector means to carry out the effect. Link that with efferent, the outward effect. The autonomic response is tied in with our sensory neurons. And it is, so let's kind of go through the process here. So it's tied in with our sensory neurons, which includes, which is choice B here. So anyhow, let's run through and continue. Okay, so one thing I'd like to address right off, and any of you who have taken anatomy might already know this, but anatomy is not a prerequisite for physiology. So let's go through. What actually is the difference between, or what is a nerve, and where are they located? So I've been talking about the afferent nerve pathway and the efferent nerve pathways, but here we're looking at an example of that highlighted in yellow. So we've got the sensory nerves, which are all distributed throughout the skin, throughout the dermis layer of the skin. But a nerve is simply a bunch of axons. So this is a neuron. A nerve and a neuron are definitely not the same thing. This is the typical cell of the nervous system, the neuron. And they have very specialized parts that serve different functions, which we'll review. But a nerve is built from a bundled sheath of axons, which is the tail end of the neuron. Okay, so looking here, we're going to identify the major regions of this neuron. So the axon is the long extension here. It's wrapped with myelin in this case. That's what these white insulating, kind of looks like hot dog buns are. So the axon wrapped in myelin. There are breaks in the myelin. This is the cell body. Here's another cell body of a separate neuron. We've identified the axon. The axon hillock is the region where the cell body feeds into the axon. The dendrites are they're really the receiving part of the next cell in the process. So these are the extensions. And we will see a similar structure with one of the immune system cells, one of the white blood cells, that's called the dendritic cell because it has a similar morphology. It has a similar shape. So these extensions, just like we see anytime in biology when we see extensions of a certain structure, it's a way to make whatever function that structure does more expansive. You can think of it analogous to increasing the surface area to volume ratio because, well, I mean, it's not analogous. It is doing exactly that. There's very little, mass or volume associated with the dendrites when you compare the rest of the cell. Okay, I think we went through all of them. So in terms of the progression of a signal through the nervous system, it begins by being received. So we're talking about coming into this cell. It's received by the dendrites that then process that information is sent in to the cell body. So this is where the nucleus and all that endoplasmic reticulum, everything to all that stuff is located. Keep in mind that we will see these dendrites can send different signals all at the same time into one cell. It's just like a bus station, right? Different pathways going into the same busy hub. But then the cell responds by sending a message out the axon down to the axon terminals into the presynaptic cleft. That's the area in between the first cell and then the receiving cell, where it's then picked up by the receiving cell's dendrites. So as we follow the progression of colors, you can match it up with the navigator here that is used throughout all the figures in the textbook. These come out of a slightly earlier edition of your textbook, but it's the same textbook. It's very similar. The reason I put these in your slides is simply because I can get just this part of the image by itself, where I can't do that with the current version of the textbook. I have to put a bunch of other stuff in there. I can't break the figure down. Okay, so I'd like you to go through and label all of these different structures that we've gone through. We've actually just reviewed all of them with the exception of the Schwann cell. And the Schwann cell is actually associated with making myelin. So we'll get to that in a moment. But please practice doing this in your studying. Okay, so where does the presynaptic neuron axon terminal interact with the postsynaptic neuron? This is a question that we were just discussing a moment before. So if we're talking about a synapse, that is the relay of information over a physical space between one cell and another. It is not always nervous system to nervous system. It could be nervous system to muscle, okay? But regardless, a synapse, the synaptic cleft is a physical space, and electrical signals go through there, chemical signals go through there. We're going to start to appreciate really quickly the role of diffusion and active transport and the variety of transport mechanisms and channels specifically that are involved in these communication cycles. Okay, so here we go. Now we've got it all labeled for you. So we've got our dendrites, the extensions. That is an increase in surface area to volume to give more of the ability to interact with the environment. Neurons do a great deal of their job by knowing what's happening in the environment. That means they're communicating with other cells in the environment. So if they have more extensions and processes to do that, then they're interacting with more of the environment. Okay, so let's focus on that synaptic cleft down here. So the presynaptic axon terminal is this area, and then we've got the cleft. And then the dendrite, it belongs to the postsynaptic cell, whatever type of cell that is. So we've got our input, we've got our integration, and then our output. So we've been talking about the afferent, the stimulus through the afferent to the integrating center, out through the efferent to the response, as a reflex through the body involving the nervous system. But we can even apply that right here to the neuron itself. It's actually a really good application and an example of the structure and function. So we've got our afferent, our integration center, and then our efferent signal leading to a response in whatever cell is beyond that, the postsynaptic cell. Yeah, so you may notice from the exam when I am redundant on topics, I'm doing it as a way to communicate to you that these are important concepts. All right, now let's make it a little bit more realistic. As a signal is sent down the axon, much of this is happening by diffusion and active transport. Okay, that's easy to say, but let's back it up a moment and think about what that actually means. Let's actually back it up to the previous slide where we can look at the axon. Here it is. Look, it's got those hot dog buns, the myelin sheath around it. That is layer upon layer upon layer upon layer of actual cell. It's actually cell that does that. We'll learn about what cell type it is. So what do you know about diffusion and active transport through barriers? Well, there needs to be an opening. There needs to be a way for that material to actually move through in order to change the membrane potential and result in an outcome. But in the areas where there is myelin sheath, that does not happen. That's a very effective insulation barrier that does separate the inside from the outside. So when we're looking at this figure, we need to keep in mind that that location, again, is important. You've heard me say that before, location, location, location. This doesn't just happen all over the axon. The signal has to be effective enough really to bounce it from this area to the break, and then from the break in that myelin sheath down the axon to where there's another break. Because it's only at the sites where we have break in the myelin, those are the sites where diffusion and active transport can occur. And as you're learning, the concentration and distribution of ions is what it's all about. So those are the areas where the ions move. Okay, so now let's kind of back up and go through the specifics of this figure. So we're actually beginning with the synthesis of proteins that are made here in the endoplasmic reticulum. So it would be the rough ER, right? If we're talking about proteins where the ribosomes are. So these are proteins that are destined to be released. When we're talking about proteins that are made on the rough ER, as opposed to with a free ribosome, it's all about, it's about secretion versus keeping the protein inside the cell for itself. So proteins that are made on the rough ER, they are sent where next? You know this pathway. It's the endomembrane system. So we actually have a direct connection from the nuclear membrane into the ER. That's not shown in this figure. It's actually inaccurate because there should be a direct connection in membrane, because there is. That means that material can move out. So such as messenger RNA, because what do you need to make protein? You need the message. So the messenger RNA can move out into the rough ER where it makes the proteins, which then pop off in a vesicle to go to the Golgi apparatus, which is next in the pathway of the endomembrane system. The Golgi processes that protein And that's the job of the Golgi. It functions like the giant warehouse, the giant post office that receives everything and then sorts it and figures out, okay, you're this type of protein, you need to go here. You're this type of protein, you need to go here. And then it sends it out through a secretory vesicle that then can move out. But we can also see the pathway of it moving down this long axon, which is what happens, because what is released at the end of these long axons at the terminals? This is where the neurotransmitters are released, and these are actually chemical proteins that are released into that synaptic cleft. So our proteins are made in the rough ER. They're packaged up and sent on their destination by the Golgi apparatus. They then move through a process called fast axonal transport, and then they get down to the terminal where they're released. So let's back up to that fast axonal transport. Okay, that means essentially it means it moves fast down the axon. I hope you can figure that kind of stuff out from the words that are given to you now, since we spend a lot of time focusing on the meaning of scientific nomenclature. So looking back at the anatomy here, the benefit of having all that myelin is it helps to send the message quicker because it insulates it. It means that it can move down without having interactions with whatever else is in the environment, because there's nothing in the environment. So that's the benefit of the myelin. And if we look at a disease like multiple sclerosis, that is one where the myelin is attacked by the immune system. People with multiple sclerosis have a lot of challenge or may have a lot of challenge with movement with their muscle control, because they can't get enough message down their long axons to send the signal to the muscles to respond. So there's no signal sent in the first place. So the protein or peptides, proteins are moving down this axon, and then they're released as neurotransmitters into the synaptic cleft by the process of exocytosis. But our body does try to recycle as much as it can, especially when we're talking about proteins and peptides, which are big and expensive for the cell to make. So it gets them back. It doesn't just, it needs a gradient, right? We're going to learn, this happens through gradient. The molecules go through that synaptic cleft based on a gradient. So the more that are secreted at the same time, the bigger the gradient, which makes it go faster. So there's a lot of different neurotransmitter released here, but they get picked back up and recycled. So they go back down the axon, the reverse direction, where they then are either digested by lysosomes, or they can even be sent back into the Golgi apparatus and so they can be packaged up and resent back out, as long as the protein is, or I should say the chemical peptide here is still functional. Okay, so we've been talking about the neuron. Now let's move into the glial cells. And these are all support cells for the neuron. That's all glial cells are. It is a category of cells that provide different support mechanisms for neurons. So the Schwann cells are part of the peripheral nervous system. They have the same function and similar location in where they're found on the cell to oligodendrocytes, which are found in the CNS. So we do have some cells that serve the same function in the peripheral nervous system as a cell in the central nervous system. And then we have even more cells that support the central nervous system, because gosh, it's really that much more important since it is where the central nervous system is. So what Schwann and oligodendrocytes do is they both make myelin. So we have two types of nervous system cells that make myelin. One makes it in the peripheral system, the Schwann cell, and then the oligodendrites or dendrocytes make it in the central. So they are actually, that is what myelin is made out of. It is these cells. You can see the nuclei of these cells in the myelin. The holes in between are called the nodes of Rambier, and this is necessary so that there are locations for the ions to diffuse and actively transport back in, because otherwise the distance may be too far to get down there. So by having these gaps, think of them like booster points. They're places where the signal can get re-engaged to send it a little bit further by interacting with the environment in the exchange of ions, which is the basis of that communication. Also, we have to appreciate the role of the electrical signal, right? Anytime we talk about ions, it also comes with an electrical signal. All right, moving on. Satellite cells are part of the peripheral nervous system, and they're like a Schwann cell, but they do not make myelin. I'll show you a picture of where they are in just a moment. So if they are outside of the central nervous system, they're called a ganglion. So you may have heard that term, a ganglion. Whereas if they're inside, it's a nucleus inside the central nervous system. All right, we've got our astrocytes in the central nervous system, and we've got a few types of these. Microglia. We've got the ependymal cells, and these are important in regeneration. These are a source of stem cells that give rise to the neuron and glial cells. So now let's look at it here. This is a much easier way to look at it rather than in a list of words. So our glial cells, let's look first at the peripheral nervous system. We don't have as many. The central nervous system arguably is much more important in function and needs more protection. So in our peripheral nervous system, we have the Schwann cells that do produce myelin to wrap the axons. There are also satellite cells that are important in specifically supporting the nuclei, again, of the cells. Okay, so we've got the moving in now into the central nervous system, we've got ependymal cells, which are a stem cell progenitor for different nervous system cells. The ependymal cells are also important structurally themselves. I'll show you in just a moment, but they form different compartments, different barriers, and therefore different compartments within the central nervous system, which contributes to its diverse function. The more compartmentation, the more diversity that there is in a structure, the more jobs that structure can do at the same time with that added complexity. The astrocytes, well, look at their structure. Look at all those extensions with these giant knobs at the end. We're seeing the same structure over and over again. And it's because the structure with increased surface area to receive input signal, that structure functions. So astrocytes are incredibly important in the balance of potassium, which is needed for communication, also osmosis, and also the neurotransmitters that are present there. Our microglia, these are considered the immune system cells of the central nervous system. They are actually a modified form of a immune system cell. So as opposed to coming from the epidymal, the progenitor cell of the nervous system, microglia cells originate from the immune system lineage. But they work like scavengers. So they're constantly interacting with the environment and taking in little bits of it and processing it to see what's out there. So they respond by releasing what are called neurotrophic factors. So these are just signaling molecules, essentially, to put it simply, within the nervous system. But they also, the microglia, actually are part of the blood-brain barrier. They're there to make sure nothing gets by. They're the sentinel cells that are there to say, this is a very important barrier. You're not getting through my army. You're not getting through my wall. Microglia also are important in the production of ATP. And then that leads us to, my mouse disappeared again. That leads us to the oligodendrocytes, which are comparable to the Schwann cells. They produce myelin, although the oligos are located in the central nervous system and the Schwann cells are located in the peripheral nervous system. So we've got a diversity of structure and function. Oh, the battery in my mouse just died. That's no problem. Okay, so now let's look at the locations of where these different cells are found. We've got, first of all, let's look at the division of color. So we've got zooming in to, we're looking in the spinal cord to begin with, and then zooming in, we're looking at an area of different sections of the spinal cord. So we've got our, where's a good place to start? Well, let's begin up here with the epidermal cell. So it is interacting with a number of different cell types and structures present here. Again, it is a source of the neural progenitor cells that give rise to the other cells of the nervous system. We've got an example of a microglia cell right here. It's the smaller cell. So these are important in the immune response and helping to form that blood-brain barrier. We've got just went through that. We've got what are called interneurons, these blue cells. And we haven't talked about those yet, but these are really important cells in connecting the cells of the immune system or nervous system. You can see the long axon here, and the cutaway shows the layers of myelin. So these are actually oligodendrocytes that are wrapping around the axon, creating that myelin sheath. And then we've got the analogous cell that does this. We've got a capillary. This may not look like a capillary, but it is. It's got vascular tissue surrounding it. We'll learn about this when we move into the blood system. So those are the main players of the nervous system. These are specifically the glial cells that provide support to neurons. Look here at the cell that's producing the myelin. So you can see the nuclei of this nucleus of the cell that is associated with wrapping around that axon. Okay, so the Schwann cell puts myelin at a space. The hot dog buns, if you will, they're not randomly sized, they are spaced. So they tend to be about 1 to 1.5 millimeters in length, and then they have a specific space in between them. The nodes of Rambier is there. So it's not random that the space is, or the length of the myelin is consistent. It has to do with the size of the Schwann cell itself that wraps around to form the myelin, right? Cells have an average size. Every cell has its average size. So this Schwann cell has an average size of 1 to 1.5 millimeters, which is why the myelin sheath comes in increments that are the same length. Okay, so looking at how that happens, the axon is in blue, and we can see one Schwann cell here with its nucleus, and it wraps it like if you've ever made a jelly roll, it's the same process. So we're wrapping and wrapping and wrapping and wrapping, winding that myelin around to where it becomes several layers thick. And the thicker the myelin, the more insulating quality that it has, which can relate to or equate in the signal being sent quicker, because it doesn't have as much resistance as the signal moves down long distances. So myelin is really multiple layers of just the cell membrane of that Schwann cell that's found in the peripheral nervous system here. We're talking about the peripheral nervous system. Okay, so now let's tie this into the communication. So let's remind ourselves about the resting membrane potential. We talked about this a little bit before. That was the topic of chapter 5, but now we're going to learn about other types of potentials through the nervous system. So we've got our graded potential and then the action potential. So we'll review resting membrane potential as well. All right, we're looking at the typical cell because of the distribution of the ions, the way they are, where potassium is higher in concentration inside the cell, and then everything else is higher outside the cell. So sodium, sodium chloride, and calcium. Proteins are higher inside the cell, and they have a negative charge. So because of this chemical imbalance on either side of the cell membrane, what happens is we have a net negative charge that builds up right inside, physically right inside the cell membrane. And then because the extracellular environment has an excess of the cations, the positively charged molecules, they come because they are attracted to the negative inside of the cell. So it again is that opposite charge interaction that is what establishes the resting, or helps to establish the resting membrane potential. So the inside of the cell has more of the anions, the negatively charged, where the outside is cations that are positively charged. So as you know, overall, our body is electrically neutral, but we do experience constant changes at a cellular level. And we need this chemical disequilibrium between the intracellular and extracellular environments, right? It doesn't work if we don't get more potassium back inside the cell. We don't get back to steady state if there are problems, okay? So the steady state is that default state, and we will look at what happens with different changes as we progress through. So this all ties into establishing the electrochemical gradient, right? So the electrical part is associated with the changes in the membrane voltage as a result of the chemicals, the ions. To me, it's very challenging to separate out the electrical separate from the chemical. And if you've read your textbook in this chapter, they spent a fair amount of time doing that, but they aren't separated. They're a consequence. I mean, the ions are charged, the chemicals are charged. That means there is going to be an electronic consequence. So we can measure this in the lab with some basic equipment, and this is what we would be doing in the lab. This is also what happens with the simulation that you see through that BioPack instrument. Okay, now that resting membrane potential is highly due to potassium, and there's a couple reasons for that. It is true that potassium is higher concentrated inside the cell, and that is one of the reasons. But there's some other reasons as well. Cells are much more permeable to potassium than they are to the other ions. They're about 40 times more permeable. So that means there's an opportunity for more potassium to move than other ions. So let's run through how the voltometer works. So here we've got an artificial cell that is in a saline bath. There are two electrodes. One is going into the cell and the second is going into the bath. So the way that the difference in charge is, so the inside of every cell is not minus 70. It doesn't work that way. What it means is that there is a difference of 70 millivolts from the extracellular to the intracellular environment. So the bath, the environment is set at 0, and then the cell is at minus 70. And the probe sends information into a chart, into the voltometer that measures the electrical current, and then it comes and is printed in a chart recorder. And we end up with a printout that would look like this. So it begins at -70, which is the resting membrane potential, and it goes along for a period of time until a period of time in which the voltage drops. It becomes more negative. So it is hyperpolarizing. Polar means a spectrum. So it's hyper, it's moving away from where it was. And then it recovers. So this period of recovery is called a repolarization. And then it moves along. And then we get to the depolarization where the membrane potential increases. And then it repolarizes again. So we've got a couple different terms that we need to understand here. A hyperpolarization is when the membrane potential becomes more negative, whereas the depolarization is the membrane potential is becoming more positive, even though the number may never get into the positive value. Those are just because of the chemistry of the membrane potential. Your book labels the repolarization here following depolarization. It does not label it here following the hyperpolarization, but both of those events are repolarization. A repolarization is when we get back to resting. So it can come from either direction. It's just getting back to that steady state. So we can look at the EMG as a way to study changes such as ion concentration, or as we saw, we will study the effects of fatigue and different variables on our muscles, which comes through the nervous system. All right, so now let's look a little bit more at that resting membrane potential. We really need to be able to understand what it is, why it occurs, and why changes occur, how to get back in order to understand what it is. And that means that's happening at the molecular level, not even at the cellular level. All right, we need to look at it even smaller than the cellular level. We need to look at the level of our molecules, specifically those ions. So we're going to begin up here with panel A, and this is at resting membrane potential. So inside the cell is more potassium. So your book is keeping it very simplified by just showing the ions as 100% in one place or the other. In reality, that's not how it is at equilibrium, but it's very close. So potassium is high inside the cell. Proteins, which have a negative charge, are high inside the cell. Sodium and chloride are high outside the cell. And this is what we are measuring in that previous example with the voltage meter. But what happens if a potassium molecule moves? So when even one potassium molecule moves through its leak channel, that's a diffusion channel for potassium, and we do have a gradient, so potassium is going to move. What happens then? Okay, now we've moved some of the positive outside, and that makes the inside even more negative because we've taken a positive potassium ion out. So now we've established more pull on the gradient. So now what happens, because this positive came out and it's more negative inside, that's going to pull sodium in. It's going to say, okay, we've got to try to balance that more negative charge. So potassium is going to lead to the attraction of, or a set of consequences with other ions when it leaks out. So our potassium is leaking out because there's a gradient. But then what happens, because we have that negative potential, it's like, okay, well, we've got sodium, we've got potassium that we could suck back in to balance out that negative. But we need to understand the dynamics of our different channels and pumps that do this. So this is what happens with the resting membrane potential. So let me just complete the bottom part of this as it's drawn here. So by having that potassium move out, we generate an electrical gradient that pulls the potassium back in the cell. So by having the potassium move out, we're putting one more positive outside, but that's the equivalent of a negative inside. So we get that pull to maintain the gradient, to follow the gradient. Now we need to understand the sodium-potassium pump. What we've been talking about was the potassium leak channel. We also have sodium leak channels and chloride leak channels that use diffusion. But these are, or I should say, use facilitated diffusion. Now we're talking about an active transporter. The sodium-potassium pump, anytime you see pump, that means active transporter. It is also why potassium is really important in establishing the membrane potential. So we'll run through all those reasons in a moment. But it has to do with the ratio of the amount of potassium and sodium that are moved. Your book is showing it even here, and we'll see a later figure that shows it more accurately. But we've got sodium that diffuses in through its diffusion channel, and then it's pumped back out through the sodium potassium pump. Same with potassium, just opposite direction. So it moves from the inside of the cell out and then back in through the pump, right? Because what happens after diffusion? Well, then we have no membrane, we have no concentration gradient. Well, it's not going to diffuse back in, it has to be pumped back in. But the amount of sodium and potassium that goes through this pump is not equal. It's actually three sodiums for every 2 potassiums. It's not a 1-to-1 ratio. So how do we change the resting membrane potential? A big effector is this pump. It gets everything back to resting membrane potential so that it may start out again using diffusion. Otherwise, the process would just end because there's no gradient. We do have a different concentration of anions versus cations in the intracellular versus the extracellular environment. So inside the cell is more negative, outside the cell is more positive. So right there we have a difference in electrical potential across a membrane that is going to result in action. And then we've got our equilibrium potential for potassium. And this is due to the fact that cells are much more permeable to potassium than they are to other ions. So I'm hoping that if you see this on, and let me back up. The average cell is about 40 times more permeable to potassium than it is to other ions, because it has more sodium channels and mechanisms for recovering. So if you see something like this on an exam, I would hope you'd be able to list them, but if I ask you to explain the mechanism or concept behind them, it's pretty elaborate, as you just saw from what I went through. So there are 3 sodium out for every 2 potassium in, and this uses active transport. And it is primary active transport because an ATP molecule is used for this. So we've got our difference between primary and secondary active transport. Primary directly uses the ATP. Secondary is once a gradient has been established by using ATP. So something benefits. Something else benefits. We've got the difference in the anions and cations on either side of the membrane, and then the differences in the permeability. So there are 75 times more potassium leak channels in the cell membrane compared to sodium or chloride. When we look at potassium as a whole, the cell is about 40 times more permeable to potassium than it is to the other ions. So these are the answers to, let me back up, to these concepts. All right, so now let's actually look at some numbers. So we've got sodium, chloride, calcium, and then potassium. We've got our extracellular and our intracellular fluid, and then the ion potential at 37 degrees for each of the individual ions. We know that potassium is higher concentrated inside the cell than it's outside. So we have 150 millimolar inside the cell versus just 5 millimolar outside. Sodium's higher inside the cell. Chloride's higher inside the cell. Calcium's higher inside the cell. And these are the individual equilibrium potentials for each ion. So potassium is minus 90, sodium positive 60, chloride minus 63. But when we're looking at the overall resting membrane potential for a cell, it's not based on just one ion, right? We've got to factor in the concentration of how many of each of those individual ions are present where. And don't worry, I'm not going to ask you to do that. There's no reason to make you do that math. That's just, sorry, this is not, I don't even know, is that algebra or is that calculus? It's math beyond what I want to grade. And frankly, I'm more interested that you understand the concept rather than being able to enter a bunch of stuff into your calculator correctly. But what is the concept? That we have an equilibrium potential associated with potassium out of the cell as well as in the cell. We have an equilibrium potential associated with the concentration of sodium outside the cell versus in. Same true with chloride. All of that together to the 61st log gives you the resting membrane potential. Again, the resting membrane potential is the difference when we set the environment to 0. It doesn't mean that it's always minus 70 in these cells, but when we set the environment to 0, as we saw in that example with a photometer, the resting membrane potential is the difference. When we go up, when we increase in potential, it is a depolarization. Then we repolarize. We're tracking along at resting. Now hyperpolarization, repolarization, tracking along at resting. And now the cell can fire off the next wave of communication because it has gotten back to its resting membrane potential. All right, so that now brings us to the next two types of electrical signals. We've got our graded potential and then the action potential. And these are signals that are specific with the nervous system. Now, a graded potential may lead to an action potential. In other words, an action potential can't happen without a graded potential. Graded potentials vary in strength though. So you have to have enough signal to result in the outcome. If you don't have enough signal, no outcome. So these are only effective for short distance because there are not mechanisms to facilitate a long distance communication. The action potential is different. It is used for the long distances, and it actually begins with the graded potential. So these are very, very quick, and they are used for long distance communication. So by sending it down an axon through a nerve often. All right, so here's a side-by-side comparison table of the differences between the graded and action potential. So you've got where it occurs, the type of ion gates that are involved, the strength of the signal, and then the unique characteristics. So I'll let you refer back to this as you need. Okay, so let's look at the graded potential. So we're looking at a stimulus. So it's coming in on the presynaptic neuron through the axon to the axon terminal. And then whatever stimulus is sent to the dendrites of the postsynaptic neuron. So in this case, it involves a sodium message. We'll look at why, but we'll keep it simple now. But what happens is the message can actually dissipate. So the bigger arrows mean there's a stronger message. But as we move further away from the exact point where the stimulus is received, the message dissipates and becomes weaker. A lot of it leaks out of the cell. There's no myelin here. This isn't an axon. So there's not a way to more effectively concentrate that signal into one area of the cell. So it may be that there is enough signal, it may be that there isn't. But as we move away from the stimulus point, looking at the graph up here, the signal dissipates in both directions. So graded potentials are really good for short distances, but they're terrible at long distances because the signal's usually gone by there. All right, so looking at two types of graded potential, we've got a sub-threshold versus the supra. So now let's look at that distance relationship a little bit further. In the sub-threshold graded potential, there's not enough. That's why it's called sub-threshold. So we have our stimulus that is received, and we see there are 3 different voltage meters inserted in this postsynaptic neuron. So we have one up top by where the message is first received, the stimulus location. Then there's another one in the cell body, and then we have another one right at that axon hillock. I like to think of the axon hillock like the armpit of the axon. That's kind of what it reminds me of. And there is a good signal. It goes up enough. If we get above this -55 line, there's enough signal to result in a signal, enough strength to result in a signal. So there is enough here. That's fine. But down here at the cell body, we're only getting to threshold. And then by the time the signal gets to the necessary point to result in an action potential, there is not enough signal. We are below threshold. So that is why this is the sub-threshold graded potential. Compare that with the supra-threshold. Now we have enough signal going through the axon hillock, through the trigger zone. Once it passes the trigger zone and becomes an action potential, it's going to happen as long as the physiology is normal. Graded potentials are different. You have to have enough to get there. But once you get there with the action potential, because there's myelin present and because we have the hexagonal transport mechanisms, the signal gets down to the end, resulting in the release of the neurotransmitter with a supra threshold. So an action potential is really like what happens with dominoes. They're a good comparison. So if the dominoes are set up perfectly spaced, they will fall with a force that is equal all the way down. So if we are to measure the strength of that force, it would oscillate up and down and up and down and up and down and up and down, but it's the same high and it's the same low. So once we get through that trigger zone, unless there's damage to the anatomy, there is going to be conduction of the signal, because at that point, the conduction of the action potential has been reached. All right, so let's run it now through what we would be doing in lab. So we begin as we're measuring the action potential. It begins at -70 at our resting membrane potential. If we get above -55, that is the threshold. So at that point, the signal is enough, and we go up to where we hit our highest point and then decrease. So we're repolarizing. So we depolarize and repolarize. So now let's though tie it into the movement of the ions that are happening. We've got, first of all, at point 3 here, what's happening is the different ion channels are opening at different times. So the sodium moves first. Remember in the figure we saw sodium with the figure with the graded potential. So the sodium channel is open, and that allows for sodium to move from the outside of the cell in, following its gradient, until we get to the highest point. And then the sodium channels are going to close and now open the potassium channels, and potassium is going to diffuse following its gradient. And that brings the potential back down to where it goes into a brief drop. Notice it's lower than resting membrane potential here. So it goes into a brief hyperpolarization, and then it repolarizes. And now all of our ion channels are reset. So if we look at the movement of the individual ions below, sodium goes first, as we can see here, and then it drops off. And then then potassium picks up, and then it drops off. So we've got the individual concentrations, the permeability associated with each of the individual ions, comparing it to the total membrane potential shown by this voltage. So it is simply changes in membrane permeability that facilitate the conduction of an action potential. And they do jump from node to node to node along the length of the axon. This is here for you to label. So you have a blank EMG graph to label. All right. So the action potential operates under positive feedback. Remember that most of our mechanisms operate under negative feedback, especially those associated with homeostasis. So this is positive feedback. Let's run through it. So we get our stimulus that triggers the response, leading to the activation of those sodium channels. So they open and sodium rushes inside the cell. And that leads to more depolarization and more depolarization. It isn't until the potassium channels open and potassium leaves that, that we see how the potassium and the sodium are under different channels. But in order to stop the sodium signal, a gate has to change, it has to close. And that happens when we get to a certain voltage, related to when the potassium gets us to that voltage. So the opening and closing of many of these gates is tied to the potential of the cell at that time. So to stop the cycle, we need something else to stop it. So the closing of the gate under a different control mechanism to stop it. Something outside of the system is necessary to stop a positive feedback loop. All right, so putting this together, we're looking now, we're getting more complex. So we're looking at the cell membrane in the top part of the figure and the various different channels that are present. So we've got our potassium channels in purple, we've got our sodium channels in yellow, and then we are looking at different time points in the permeability of this membrane. Sorry about that. I lost the screen share there. All right, so we've got our different channels, as you can see, and we'll track the movement of molecules through them over the progression of time. So on the x-axis, we're looking at time, and it is in milliseconds. And then on the y-axis, well, depending upon what part of the graph we're looking at, we have different scales. So we have the sodium and potassium channels in this top row. In the middle row is membrane potential. And then in the bottom is the excitability. And then here we're looking at, in the pink area, is called the refractory period. We have two types of refractory periods. The pink is what's called the absolute refractory period. The purple is the relative. The absolute refractory period is a time in which the cell is unresponsive to the next stimulus, because the membrane potential is somewhere else. The ions are not back in their right place. There's not a way to send the signal. The relative refractory period is different. The cell can respond, but it may or may not need more signal. It may need more signal, or it may need less signal, actually. So problems can happen when a cell's responding during the relative refractory period. So if we look at the membrane potential, we saw this in just a figure before. We've got the action potential, so that's the voltage of the cell, of the membrane. And then we've got the graphs for the sodium concentration and movement and the membrane potential associated with it versus the potassium. So the period of absolute refractory period, the sodium channels are closed, our potassium channels are open. And as the potassium flows from inside of the cell out, this is helping to drop the action potential back down to where it hyperpolarizes and eventually gets back to normal. In the relative refractory period, our sodium channels are going back to normal and the potassium channels are still open. And then we get back to a period of time in which they are both closed that precedes the firing off of the next potential. So we do have a difference in the absolute refractory period versus the relative refractory period. The cell is responsive in the refractory period, but it's going to respond differently. In the absolute refractory period, a stimulus is not capable of doing anything under normal conditions. And then if we look at the excitability, it's increasing, right? There's none during the absolute refractory period because nothing can happen, but it does increase through the relative refractory period. So in terms of the time, it occurs between 0.3 milliseconds to 4 milliseconds. And we are talking about a neuron, right? We're in the neurology chapter. So we're talking about the refractory period for the neuron. We have, again, the absolute and a summary of this, what's happening with the gates here. We just went through that. And then we have that relative. So some of our channels have reset, and some of the gates have reset back to the normal concentrations, but we need a stronger than normal stimulus to hit action potential, right? If you're refractory to something, it means that you're unresponsive to whatever that is, or you need more of it to respond. Okay, so I've given you some more terms to now associate with these different images. So please take the time to not only learn what these words mean, but understand the concepts and then be able to apply them and label them onto a graph, or even draw the graph yourself completely, please. Okay, let's go ahead and play this. It's not too long, it's just 4 minutes, but it's a good one. Oh, just a second. I need to stop and restart the share. I've learned that.
Biology is really simple. Everything in biology works based on a few basic principles. The action potential is a really good example of this. It might look pretty complicated, but all you need to know is how diffusion and active transport moves ions. This is the structure of a myelinated neuron. And if we look closer at the axon, we can see that when the neuron is at rest, there is a higher concentration of sodium ions outside the cell compared with the concentration inside. And it's vice versa for potassium. Because of the concentration gradient, an active process is needed to maintain a situation, the sodium-potassium pump. This uses energy in the form of ATP to move the ions against their diffusion gradient. And this gives us a resting potential in the neuron, which is about minus 70 millivolts, and it's said to be polarized. Now we're going to look at what causes the pattern of changes in membrane potential when the neuron fires. Looking at the soma of the neuron, we can see the dendrites. This is the site of synapses with neighboring neurons. The impulses received are processed, and small amounts of depolarization spread to the axon hillock. If this small amount of depolarization reaches a threshold, it causes voltage-dependent sodium channels in the axon membrane to open. Sodium ions diffuse along their concentration gradient into the cell, causing the inside of the neuron to become positive compared to the outside. This stage is represented in the uphill part of the action potential trace. The depolarization from the soma reaches threshold and opens the sodium channels, and this allows a rapid influx of sodium ions. This stage of the action potential is called depolarization. Looking back at our neuron, the sodium channel is now closed, and potassium channels will allow potassium ions to diffuse out of the cell. This swaps the polarity of the membrane again, so that the inside is now more negative than the outside. And we can see on the graph that depolarization generally reaches around plus 40 millivolts before these potassium channels open. As potassium ions diffuse out, this causes repolarization of the membrane as the inside becomes more negative again. Potassium channels open and close very slowly, meaning that too much potassium diffuses out, resulting in hyperpolarization. This is then corrected because sodium and potassium leak through the membrane. This is by diffusion with their concentration gradients. The sodium-potassium pump also helps. This brings the membrane potential back up to the resting potential. And you can see this in the graph too. In the final part, potassium channels close and the hyperpolarization is corrected. This restores the resting potential. The time when the membrane is hyperpolarized is called the refractory period, and during this time, no action potential can occur. And this also means that the action potential always travels in only one direction. Remember, we've been looking at only a small section of the axon. The nerve impulse is propagated because these events occur repeatedly next to one another. The depolarization during the early phase of the action potential travels along the next part of the axon. This opens voltage-dependent sodium channels, and the process repeats. This is how the impulse is passed along the length of the axon. Remember also that in a myelinated neuron, like the one pictured here, action potentials only occur in the nodes of Ranvier. These are areas where there's no myelin insulating the nerve, and it produces faster conduction as the impulse can jump from one node to the next. The action potential is really simple. All you need to understand is diffusion.
All right, let's get us back now to your slides. We're getting there, almost done. Yeah, that video is great because it summarizes many of the concepts that establish resting membrane potential. So conduction happens through the length of the axon sheath, or even through a nerve, because there is the potential for diffusion and active transport to happen with the ions. And that happens at these active regions. So this is where there are gaps in the myelin. So we see that the dynamics of how the electrical current changes, it feeds into what happens next. For example, if we're in this active region at this time, we'll just start there where we've got sodium moving in. Sodium's positive, so we're going to have a positive, a buildup in the charge from the influx of that positive sodium. Because the region right next door to the sodium is negative, that sodium is going to move over to it's going to be attracted to that negative charge, driving it in a way to where the active region is. So as it drives it this way, it's taking it into an inactive region. As it drives it this way, it's in that refractory region where it's not able to respond or needs a stronger than normal signal to respond. So this happens as the signal moves down the length of the axon, and it makes it so that the signal flows in one direction as opposed to, going back and forth different directions at the same time, which would be very ineffective. So the refractory period here, it prevents the system from going backwards, right? It's active, but it's going to keep it moving towards the inactive region. The inactive region becomes active as the ions and electrical current changes. So it has a lot to do with the potassium again, because that refractory period is tied heavily to the fact that potassium is going to move. So it's able to repolarize the membrane because those potassium channels are still open, but the sodium ones aren't at that time. So this is called salutary conduction, and now we can see that this happens at the nodes of Ranvier, not where the myelin is. And so the signal is able to be passed down because in the areas where the myelin is, it's insulated. So there's no loss of the ions, there's no loss of the electrical potential. That's only happening as the signal moves in a controlled direction, one direction down the length. But when we have demyelinating conditions like multiple sclerosis, where the myelin is compromised and broken down by the immune system, now there's the opportunity for the signal strength to dissipate because it has the opportunity to diffuse out or move out in areas that otherwise would be blocked. So this is why action potentials, once they are generated, providing we have our normal physiology, they do result in their response, because the system is designed to do that. But when our response is compromised by a pathology, then you can maybe begin to understand why people with diseases like multiple sclerosis have limitations in their ability to move and even talk or eat, because they can't control their muscles. They would need a way stronger signal strength to have the same outcome than a healthy person would, because it would need to be so strong that it can have enough even when it diffuses and dissipates out. So myelinated nerves do conduct a quicker communication. All right, so now let's run through different conditions. We're going to look at the conditions of different potassium concentrations on the resting membrane potential. And we know that potassium is really important. So here we're looking at normokalemia. Kalemia is potassium. So normokalemia is when the potassium is in the normal range, but we've got a sub-threshold stimulus. So our stimulus is applied at this time, and we see an increase in the resting membrane potential, but it's not strong enough. This is a graded response. It has not hit threshold. So therefore, there's no response in this panel. But now we're looking at a person that still has a normal potassium level, but now they get a strong enough stimulus to pass threshold. So there is an action potential fired, because all they have to do is pass this -55 level threshold, and then it happens. But in the situation when there is too much potassium in the blood, remember potassium is supposed to be higher in the cells. So in hyperkalemia, there's too much potassium in the blood. So that is going to take a positive ion and put it in the blood. So we're going to be more positive. So now we're even closer to threshold. So in the condition of hyperkalemia, we don't need as much stimulus. A smaller response can get us above threshold because there's less area to the threshold. In hypokalemia, now there's too little potassium, and people with hypokalemia have a lower resting membrane potential, or they are at a value lower than resting membrane potential. So they're down here at, say, -90. So they have the normal size stimulus, but it does not get to threshold. So they're not able to fire the action potential. So a person with hypokalemia often is tired and fatigued, and they literally are. They don't have the capacity to fire the potentials that are needed to move those muscles. It's not that they're just being lazy. So potassium is a very critical electrolyte in the function of skeletal as well as heart muscle and smooth muscle, and it's important to keep it within our healthy physiological range. So we do have a number of mechanisms for that. So looking at a zoom in now of the synaptic cleft, we've got our axon terminal, and then inside these vesicles are the neurotransmitters. There's lots of mitochondria to provide the energy to move all these vesicles. So what happens is it is the action potential itself that moves down the axon terminal that causes the depolarization, causing the calcium channels to open. And now calcium is going to flow from the outside in, following its gradient. When calcium does that, it causes the synaptic vesicles containing the neurotransmitter to release. And now we have the neurotransmitter that diffuses following its gradient into the synaptic cleft, where it will bind to its match protein receptor on the postsynaptic neuron, leading to the response. So a lot of this is similar to any other receptor-mediated response. The cells that have the receptor are able to respond. So if the neurotransmitter binds to the receptor on the postsynaptic neuron, what next happens? So I'd like you to run through the steps that we just went through and be able to answer any question about any of these steps. So we're talking about this response. So once the calcium is released, it binds to the receptor by moving through the synaptic response, and then that leads to the cellular response in whatever cell that is, the postsynaptic neuron. So once the neurotransmitter is secreted, it doesn't continue to stimulate a response because it has to be continually secreted because that gradient's going to go away. Sorry about that. Somebody just came to my door. That's one of the weird things about teaching on Zoom. Okay, so in order for the postsynaptic neuron to continue with its response, it needs more neurotransmitter. The thing I haven't shared with you yet is we need the gradient, yes, but there are enzymes that are present in the synaptic cleft that break down the neurotransmitter. So it does need to be secreted and secreted and secreted to lead to a continual response. It's also recycled efficiently. So how do we maintain that constant stream? We need to fire more action potentials from the presynaptic neuron. What will cause an increase in the amount of neurotransmitter? Well, more action potential. Okay, or an additional action potential will release will lead to the release of more. So here we have showing how neurotransmitters are recycled. So we can have them picked up by the glial cell or directly right by the presynaptic neuron, which then can package it right back into a synaptic vessel, providing that it still is a correct structure and has the correct function. So we see the example of an enzyme here that is digesting the synaptic cleft that is in the neurotransmitter that is present in the synaptic cleft. So it does need to be continuously released to lead to more response because there are mechanisms to clear the cleft of any remaining neurotransmitter. Sorry about that, class. All right, now this is divergence versus convergence. The primary difference is the number of cells either sending or receiving. In divergence, we have one presynaptic cell that activates a bunch of cells. So the response diverges as more cells and more cells and more cells, the same type of cell, are activated from only this one presynaptic neuron or a few. Where in a convergent pathway, it's the opposite. So here we have many presynaptic neurons that converge to fire a signal. So maybe convergence is a way to deal with a sub-threshold signal, right? If we don't have enough coming from one cell, what if we add together a bunch of signal from a bunch of different cells? And then we get enough signal to fire the action potential. That actually does happen, and that is what happens. So we've got what is called summation, and this is just the way that our cells process and integrate different information that's coming into the same cell. So in spatial summation, we have two or more neurons that are fired, and that response becomes additive. So I'll show you some examples of it. So in spatial summation, it's like convergence. The more signal that's coming in, the higher the chances that there will be enough stimulus to lead to the response. We do see inhibition as well as excitation. So I don't want you to think that every signal that comes out of the nervous system results in turning something on. We also have pathways that turn things off. So some neurotransmitters inhibit pathways, whereas others excite pathways. And if we're getting both signals into the same neuron, well, the neuron needs to figure out how to process and integrate all that information. And it does. We'll see how. Now, temporal summation is different. This is happening over a time course. That's what temporal means. It's like seasonal. So it happens when the graded potentials are overlapping in time. So spatial summation, multiple signals coming in to the same postsynaptic neuron. And if they are strong, if we get enough in, it might be enough to trigger that threshold, firing through the trigger point into the axon. Whereas with temporal summation, it is the same signal that is hitting over and over and over again. So separated in time. Temporal is time. So the same signal hitting over and over again, but hopefully if it happens close enough, it's enough to boost us up above threshold. Let's take a look at these videos.
Will the neuron pictured in the center here go to action potential? Well, in order to decide, it must interpret all of the incoming information. And this may be difficult because there may be 10,000 synaptic knobs from other neurons which contact its dendrites and soma. The neuron depicted in the center, the postsynaptic neuron, must perform spatial summation. It must add together the cumulative effects of all of these presynaptic neurons. Now, some of these presynaptic neurons are releasing neurotransmitters which depolarize the postsynaptic neuron. In other words, their effects are causing excitatory postsynaptic potentials, or EPSPs. In contrast, some of the neurotransmitters secreted by these presynaptic neurons are inhibiting the postsynaptic neuron and making it less likely to go to action potential. They are causing IPSPs, or inhibitory postsynaptic potentials. There is only one section of the postsynaptic neuron, however, which can generate an action potential. That is an area at the base of the axon known as the axon hillock, also known as the trigger zone. The only time that the postsynaptic neuron generates an action potential is when this axon hillock depolarizes to threshold. Depolarization in other parts of the cell, such as the dendrites, do affect the membrane potential but do not cause an action potential. Only the axon hillock is capable of generating an action potential. And so therefore, if the cumulative effect of all of these other neurons added together, all of their EPSPs and IPSPs, depolarize the axon hillock to threshold voltage, then the postsynaptic cell will generate an action potential. Now, not all of these presynaptic neurons have an equal effect on the axon hillock. Some are closer. And so therefore, as synaptic knobs which are closer to the axon hillock have a greater effect on whether or not an action potential is generated, while synaptic knobs which are far from the axon hillock, terminating in dendrites, for example, these synaptic knobs have a lesser effect on whether the postsynaptic cell goes to action potential. This is spatial summation, the adding together of the cumulative effects of many synaptic knobs, and position matters. The closer a synaptic knob is to the axon hillock, the greater its overall effect.
All right, so that is an example of spatial summation, and it brings in the combination of the excitatory signals and the inhibitory signals. The way I like to think about it, I like to make things simple, is like a bank account. You can put money in, you can take money out. As long as you have enough money at the end of the day to buy what you want, then you're okay. So as long as you have enough signal, when you take into account the difference between the excitatory and the inhibitory signals, then the signal goes through. All right, I'm now going to play the video, the second video that is on that slide for temporal summation. Let's see, where are we? That's it. Sorry about that.
Welcome to another episode of Interactive Biology TV, where we're making biology fun. My name is Leslie Samuel, and in this episode, episode 19, I'm going to be answering the question, what is summation? And I'm going to talk about the two types of summation. So let's get right into it. Summation is basically the addition of postsynaptic potential. To illustrate this, I'm going to draw our membrane potential graph with membrane potential on the y-axis, E sub m, and time on the x-axis. And I'm going to start with our resting membrane potential somewhere around negative 70 millivolts. So let's say this is negative 70. Now we've spoken about threshold, and we said that in order for there to be an action potential, the membrane potential needs to reach that threshold. And I'm going to say that's around negative 55 millivolts. So an action potential comes down an axon. It reaches the axon terminals. Neurotransmitters are released, and that causes an excitatory postsynaptic potential in the postsynaptic cell. What you're going to see there is there's going to be a little bump in the membrane potential. Now we know that the threshold is up here, so that bump is not going to be enough to cause an action potential. So what needs to happen is we get an excitatory postsynaptic potential, and before the first one finishes, another one comes along, raising the membrane potential even more, and that process continues over and over until eventually we have enough stimulation to cause the membrane potential to reach threshold, and then we get the action potential with our depolarization, our repolarization, our hyperpolarization, and then the action potential is finished. So that's what we're talking about with summation. We're basically adding these postsynaptic potentials. Remember, sometimes we can get excitatory postsynaptic potentials. We can get inhibitory postsynaptic potentials that bring our membrane potential even farther away from the threshold. But we're basically adding them so that we can reach that negative 55 millivolt threshold and cause an action.
So this is temporal summation. The same cell, the same signal is being fired over and over and over again, but it's happening before the cell gets back to resting membrane potential. And if it gets above threshold, then we do get summation strong enough to result in the action potential. I'm going to go ahead and continue on with the rest of the lecture, but please do go back and watch that entire video yourself, because there is good information in the rest of it. Let's get the slides back shared. Let's see, here we go. Okay, so spatial summation, we're looking at all one type of signal here coming in from 3 different presynaptic neurons. It adds together, and if we have enough that passes through the trigger zone or through that axon hillock, it results in the action potential. So the closer the input is to the trigger zone, the more likely the action potential gets fired, because these signals dissipate over distance. So the closer you are, the better. Now, as we saw in the video, some neurons are going to secrete stimulatory or excitatory neurotransmitters, where others are inhibitory. And what matters is, do the stimulatory ones win out, or do the inhibitory ones win out? What's the end of that math, if you will? Now we're looking at an example of 2 stimulatory and 1 inhibitory signals coming in from 3 different presynaptic neurons. And this time we do not have enough signal to generate the action potential. So there is no action potential fired here. So how does a postsynaptic neuron react if one presynaptic neuron increases the rate of stimulation? This is a good question related to temporal, because temporal summation is all about refiring over different time periods. I mean, it's very quick. It's happening in milliseconds, but it's not at the same time. It's happening over a time period. So let's look. We have stimulus 1 here, resulting in a sub-threshold response. Here we hit it again with the same stimulus, resulting in another sub-threshold response. But now the 2 stimuli have been moved closer together in time. And the first stimulus goes, resulting in the first response. It starts to come down, but now we get hit again with the second stimulation, which happens to be enough to get above this minus 50-fold 5 millivolts, which is the threshold for a neuron. So the action potential is fired because of the temporal summation. Without it, there would be no action potential. So summation is a way to overcome the sub-threshold responses. It's also a way to control them. Think about inhibitory and excitatory stimuli. That would be control of the response. It may not excite it at all. It may inhibit the response. So we've talked a little bit about long-term potential more in lab. And this is when we use the same neural circuit over and over again, it gets faster because of that repetition and frequency. I mean, think about learning. The first time you read something, unless you have a photographic memory, you might remember part of it. Okay, let me talk about me. I've had examples where I've read a journal article before, and I write great questions in the sideline. And not only do I not remember that I never read the journal article, I'll come back and read it again. And then I'm like, how on earth did I come up with that good question? Because I can't even remember what I read. I do not have a photographic memory. But I know how to study. I know how to develop. I know how to learn. And that is done by repeat interaction with different media types for me, and as it is for most students. So learning in school is analogous to what is happening in our body. And that is the basis of long-term potentiation. It's a process that can improve with frequency. So the more we use a neural circuit, we develop that muscle memory. What's actually happening? Well, it could be due to the fact that more neurotransmitter is being released. That could be one of the biological reasons that's leading to that long-term potentiation. So there's a reason, it's not just random. It could be that the vesicles are storing more of that neurotransmitter. That means more can be released quicker. That could mean there's a stronger gradient too. There's other reasons too. Maybe there's more receptors on the postsynaptic neuron that can deal with all of that neurotransmitter that is received. So there are a number of viable scientific pathways that have been worked out to show how long-term potentiation happens. We studied it in lab by doing the BioPack measurement, where we squeeze the diameter in our dominant hand and our non-dominant hand. The theory, the hypothesis is in the dominant hand, those muscles are stronger because they're used more. So therefore we should be able to hold it longer. And depending upon which subjects you selected, you only looked at 2, you may or may not have seen that. But if we had looked at a whole class data set, we would have seen that the dominant hand did result in the ability to hold that diameter, that hand squeezy thing, for longer periods of time at higher grip strength too. All right, that's it. We're done with the chapter 8 lecture. So that's the neurology. We're going to move out of neurology and go straight into muscles, which will be nice because we'll get back to lining up our lecture and lab content so that it pairs together well. Have a good day, a good night, whatever time it is, wherever you are, and take care. I'll see you again soon. Bye-bye.